Okay, hello everybody. So I guess we better make a start. Um, the last lecture in this series is about viral diseases and epidemics. So I'm going to start with a few basic concepts in viral infections. One of the things that will become apparent is that for acute infections, there are some viruses where you catch them once and you acquire durable immunity. And there are other viruses that you can catch multiple times over your life. And this is going to be related to this concept of what's called a serotype. So we'll look at what serotypes are. So once we've done that, we'll have a few specific examples of acute infections. So looking at poliomyelitis and dengue, and what are the vaccines available, how they work, and what are the perspectives for control or even eradication of these viruses. Then we'll go on to influenza virus and look at the concepts of antigenic drift and antigenic shift, which are basically the reasons why this virus will never ever be eliminated from the human population. And we'll end up with a short look at prion diseases, which are not exactly viruses, but they kind of come into the general category of really, really small things that are transmissible infectious agents. Okay, so for general concepts in viral infections, every infection has got to start somewhere. So the virus has got to find a way into the new host organism. And viruses infect cells, so they need to infect the first cell at an interface with the external world. And most viral infections start at mucous membranes. So either in the gastrointestinal tract, in the mouth or in the gut, you will catch these viruses by ingesting them. So the mode of transmission is oral. And for example, viruses transmitted through the mouth that will replicate in the intestine are rotaviruses that cause gastroenteritis, responsible for a lot of infections in children and even death sometimes, especially if there's not much clean water available. Hepatitis A virus, once again, it's a waterborne virus and poliovirus. So all of these three will have fecal-oral transmission. They replicate in the gut. A large amount of virus will be secreted in the feces, and if this gets into the water supply, that's the next route of transmission to the next host. So all of these viruses being non-enveloped, okay, so they need to be quite resistant, so they're non-enveloped viruses. Respiratory transmission, okay, a lot of viruses can infect us, by the airway mucosa. So influenza virus, rhinovirus that causes common colds, measles and adenovirus, all of these transmitted by inhalation. Sexual transmission, important route of infection for several viruses like the papillomaviruses associated with cervical cancer, and of course HIV and hepatitis B. Okay, what about the skin? So generally speaking, the skin is not a very good surface for viruses to infect because the external layer is made out of keratinized dead epithelial cells. So the cells on the outside of our skin, they're dead. They cannot support virus replication. So there are some viruses that can seemingly get into the skin just with a minor abrasion. This can be like some pox viruses that can infect children with a mild infection, and papillomaviruses that cause warts on your fingers or your feet. So they can somehow start an infection just with a mild abrasion on the skin. So for most viruses that kind of start infecting you via the skin are actually injected under the cutaneous layer by a vector. So vector transmitted diseases, arthropod-borne viruses, arboviruses, like yellow fever, dengue, will be injected by the mosquito, a mosquito, and tick bites can also transmit some viruses. Now, another type of, okay, so vectors that viruses that are transmitted by vectors, in some sense, they're transmitted by the blood, right? Because at some point, the virus is going to replicate, give a high viremia, so you'll have virus in the blood, and the vector comes along and acquires virus from an infected person, then transmit it to someone else. So all of these viruses here, the ones that are transmitted by vectors, they've got to have a pretty high level of viral load in the blood. And that's also true for the viruses that have kind of direct blood-borne transmission, like HIV, hepatitis B, and hepatitis C. And so these can be transmitted by blood transfusion, injectable drugs, also by some kind of like practices like tattooing, this kind of thing. If the material isn't sterilized between clients, then you can get transmission of hepatitis B, hepatitis C from one person to another. And something perhaps which is a bit more unusual is transmission by organ transplant. So that's specifically the case for herpes viruses that have the capacity to remain latent in tissues, cells of your organism, and specifically and particularly for hematopoietic stem cell transplant. So if you get like a bone marrow transplant, then you can carry over with the bone marrow infect cells that have got a latent infection of Epstein-Barr virus or cytomegalovirus, and then these can cause trouble in the host. It gets the transplant recipient. And the last one, which is very important, is mother-to-child transmission. So this can occur in utero during pregnancy. Cytomegalovirus is one example of that. Very often during birth is an occasion where viruses can be transmitted to the newborn baby. So that's true for principally for HIV and hepatitis B. And viruses can sometimes be transmitted through the breast milk. Okay, so that's true for HIV. And this is an extremely important mechanism of virus transmission. It's the main motor that is driving, for example, hepatitis B persistence in the human population. This is the main thing. So why is it important to know these things? Well, whatever strategy you're going to put in place to prevent viral infections or to limit viral epidemics is going to have to be adapted to the mode of transmission. So, you know, like wearing a condom is not going to protect you from influenza, okay? Wearing a mask is not, a face mask is not going to protect you from dengue fever, getting a dengue virus infection, okay? So understanding routes of infection, modes of transmission, essential for control of viral infections. Okay, so once the virus gets into your system, one of two things can happen. Either replication remains localized, and then you'll have no viremia, so the virus will not circulate in large quantities in the blood. Generally speaking, these infections have a short incubation period, and the incubation period is the delay between infection and when you get the first symptoms. So localized infections generally have a short incubation period. So examples will be influenza, rhinoviruses. So these will be infecting via the respiratory mucosa, and they stay there. That's where their site of replication is. They cause symptoms by infecting cells at the initial site of replication. Rotavirus, same kind of thing, causes gastroenteritis. It infects cells in the intestinal epithelium, replicates there, that's it. It doesn't go anywhere else in your body. And it's also true for papillomaviruses, okay? So if you have warts on your skin from a papillomavirus, that's it. It's the virus has infected you right here, and it's replicating at that particular site. That's it. You don't get, replication of the virus, in your liver, for example. It stays localized. Now, the other alternative is a systemic or a generalized infection. So these infections will cause symptoms when the virus infects an organ which is distinct from the initial site of infection. And to go from the initial site to the organ where the virus is going to cause trouble requires the virus to be transported through the blood. So you'll always have a viremic phase. And because you'll have probably first round of infection at the initial site where you get infected, and then symptoms later on when the virus has gone to a distant site, this results in these infections having a longer incubation period. So they're, a lot of them are like around about 2 weeks long, so 1 to 3 weeks, or even longer, sometimes up to a month. So for examples, measles and chickenpox, like varicella, these are both going to infect you by the respiratory mucosa, and there'll be something like a 2-week incubation period before you start to get a strong fever and coming out in spots all over your body. And the reason why these spots are all over the body is because the virus has got everywhere. Okay, you've got a systemic infection. So it's also true for Epstein-Barr virus and HIV. So for example, HIV, the initial site of infection will be the genital mucosa, and it's causing trouble when the virus gets to the secondary lymphoid organs and destroys a lot of the CD4 T cells in your immune system. And once again, it doesn't stay localized in the genital mucosa. It's going to get all through your body and infect all your lymph nodes and your spleen. Okay, so different types of viral infection can have different progress through time. So an acute infection, an acute self-limiting infection will have this type of process. So you'll start with the virus arrives, infects you, you get peak of virus replication, symptoms, fever, feeling sick, and then your immune system will click into gear and manage to eliminate the virus. And so typical viruses that cause these acute self-limiting infections will be, noroviruses, could also say rotaviruses, adenoviruses, rhinoviruses. So these are viruses giving you acute respiratory or gastrointestinal symptoms. On the other hand, you can have some acute viral infections that become very, very severe, symptoms get worse and worse, and you can die. And the most severe, most pathogenic virus infections will be things like Ebola virus, Zaire Ebola virus, or rabies infections. So you'll have maybe 50% survival rate for Ebola virus, and you might have more than 80% death rate for a symptomatic rabies infection. And there's a whole spectrum of outcomes in between these two. So for these viral infections, almost nobody infected will die. For these ones, you'll have a very high percentage. And then you've got things like influenza, dengue virus will be somewhere in between. So most people will have an uncomplicated disease course, but some people would have a severe infection and can die. So the death rate for these things is going to be non-zero. So those are acute infections. Viral infections can also be persistent, and persistent infections can either be latent, and latency is kind of illustrated here. So there's a primary infection or primal infection where you get the same kind of thing as you observed here, peak of virus replication symptoms, and then the virus seems to disappear. There's no active virus replication, but in fact it persists in a latent form somewhere in the body. And then periodically, months or years afterwards, active virus replication can reactivate, and you get the resurgence of symptoms somewhere. So the real classic, the masters of this are the herpes viruses. That's their big thing. They provoke a latent infection, and then from time to time you can get reactivations. So it's the case for herpes simplex, cytomegalovirus, also varicella virus. And we can compare that with chronic viral infections. So in a chronic infection, you get primary infection, some symptoms, but as the symptoms die down, in fact, the virus is not really controlled. You have persistent, active virus replication all the time throughout the disease course, and you get symptoms, might come back, might be controlled, but there'll be a tendency for these symptoms to get worse and worse, because all the way through this period of years and maybe decades, the virus is infecting cells, killing them, and the target organ has got to somehow regenerate. But there's a limit to this process. So in the end, you'll have the target organ which will fail, and that can lead to death. So this is typically the chronic hepatitis infections. Okay, so the liver has got a huge capacity for regeneration. And as Prometheus found out, right? How was Prometheus punished by the gods? Anybody know this one? Huh? Yeah. He was tied to a rock, and every day a bird would come and like eat his liver, and every night it would grow back. So it was like torture forever. So the liver's got a big capacity for regeneration. So it can, you can manage to function for years and decades, even with a persistent and chronic viral infection. But in the end, you get like progressive disease and progressive destruction of your liver. And this can lead to fibrosis and liver cancer in the end. Okay, so those hepatitis B, hepatitis C are chronic viral infections, and also is the case for HIV. Untreated HIV infection is a chronic viral infection with continuous replication all the time and destruction of your CD4 T cells. But it's also capable of resting in a latent state. So this is why it cannot be cured by antiviral drugs, because if you stop the therapy, then the latent reservoirs of HIV will reactivate, and then you get back to this like a chronic infection again. Okay, so I talked vaguely about the death rate of different infections like rhinoviruses, influenza viruses, or Ebola virus Zaire. And the way this is quantified is by numbers which are called the case fatality ratio and the infection fatality ratio. So the CFR is the number of fatal cases divided by the number of clinical cases, whereas the IFR is the number of deaths divided by the total number of infections. Now, the IFR is not easy to really know or calculate accurately, because you would have to know the total number of infections, including people who have an asymptomatic or a mild infection. So they never go to their doctor to say, oh, I'm sick. So how do you count these infections? It's not very easy. So for most viruses, what we actually know is, or have been able to measure, is the CFR. Because at least in the CFR, it's the number of clinical cases, people who've been sufficiently sick to go and get some kind of medical help. So they get registered somewhere in a database, so you can count them. And you can count the number of deaths. So the CFR, there's some data that we have for some representative acute infections. So in the biggest epidemic of Ebola virus Zaire that we've ever experienced, there were almost 30,000 cases, people who got sick, more than 11,000 deaths, and the CFR was about 40%. So this is a very, very severe, highly pathogenic virus infection. In a kind of similar category, there was historical data from smallpox, so which has now been eradicated. There's no current data for this infection, fortunately. 3,000 cases admitted to a hospital in India, 944 deaths. So the CFR about 30%. So this number is often quoted, okay? But you should bear in mind that the patient population is already people who are sick enough to want to go to hospital, all right? So it's maybe an overestimate of the, certainly not, the IFR, the infection fatality ratio, is definitely going to be lower than this, I think. So that's like going from very, very severe, and then slightly less severe down here. So I got some data from measles, Democratic Republic of Congo, 2019. There was a major epidemic, more than 300,000 cases, 6,000 deaths. So here the CFR was almost 2%. Most of these young children, of course, you know. And then for seasonal influenza, these are the numbers from the last winter wave of influenza before COVID-19. So in France, we had maybe almost 2 million cases, people who are sick enough to go and see their doctor, I've got flu, and then maybe 8,000 deaths. So, a non-negligible CFR of about 0.4, 0.5%. So, this is the kind of numbers we have for different acute viral infections. Now, the other things that are important concepts to know are the prevalence, incidence, and attack rate. So the prevalence is the number of people who carry an infection at a particular moment in time. And the incidence is a kind of rate, okay? It's a number or proportion of new cases per unit time. So these two numbers can be quite different, especially for chronic or latent infections. The persistent infections, you can have a high prevalence, but the incidence might be quite low. Okay, for an acute infection, these numbers will be, well, slightly different things, but they'll kind of go together. So for a chronic infection like HIV, in the country which has the most serious HIV/AIDS epidemic in the world, which is South Africa, I looked at these numbers just the other day. It's down to 18% now, but it's still, a very high proportion of the adult population which is seropositive for HIV. Almost like 20% of people. So just think about it, looking around, a fair few people in here seropositive if we were living in South Africa. And so the seroprevalence is pretty high, but the incidence is not that high, okay? still 240,000 new infections is a serious thing. So UK, France is similar numbers for HIV, seroprevalence less than 0.5%, and the number of new infections is actually pretty low. One more thing to say about this is that, okay, imagine if we brought this number down to 0, right? If this goes down to 0, the year afterwards, will this number go down to 0, or will this number be reduced, stay the same, or increase? It can't really increase, but it will stay the same. Why will it stay the same? Yeah, but why doesn't it go down? Because if you stop all the new infections, why doesn't this automatically go down? Ah, okay, so you stay infected and you just like persist. But why, okay, so what are the two terms that go into determining this? If you're not cured, what will happen to you? Eventually, you die, right? So the prevalence, okay, will be, it's the balance between the new infections, which will tend to increase it, and the number of, okay, cures, maybe that's 0, or deaths, which will decrease it. So this is one of the things in HIV AIDS is that the worldwide total number of cases has not really come down, but that's because more people have access to treatment and people live longer. So it's kind of a success. So the number of new infections has gone down quite a lot in the past 15 years, but the prevalence has stayed pretty high, number of total infections. Okay, so that's kind of overview of viral infections and a little bit about virus epidemiology. So neutralization and serotype. So why is this important? So in an acute viral infection, as I said, you have a peak of virus replication, symptoms, then your immune response kicks in, and mostly it's the cellular immune response which is going to eliminate the virus-infected cells. So this is the main thing responsible for clearing an infection. And then towards the end of an acute infection, you start getting antibodies in the blood. And their main role will be to prevent future infections, because these antibodies will stay in your blood for a long time. Whereas the cellular response, you get memory T cells, but there are not so many of them circulating around. Now, there are acute infections you catch once in your life, and then you acquire durable immunity. So, for example, polio, measles, smallpox, varicella, you catch them all once. And there are also recurring infections. So you can catch a rhinovirus infection or an influenza virus infection, multiple times during your lifetime. Why is this happening? Well, it's because all of these guys, they have just one or a limited number of serotypes. Whereas for the recurring infections, you have maybe dozens of different serotypes for rhinoviruses and adenoviruses. So what does this mean, this serotype idea? Well, it means that if you get antibodies to one strain of adenovirus, they will not protect you against infection by an adenovirus of a different serotype. So if you imagine this is an infection with adenovirus serotype A, these antibodies that you get, you won't be reinfected by an adenovirus serotype A, but you can get infected by another strain of the same virus. So you can get multiple infections during your lifetime. Okay, so I'm talking about antibodies, but the ones that are really going to confer protection are what are called neutralizing antibodies. So neutralizing antibodies are the ones that will bind to the surface of the virus and prevent it from entering and infecting cells. So they can block the interactions between the viral attachment protein and its receptor or its co-receptor, or they can prevent the deployment of the fusion domains. And the reason that they can prevent future infections is that some of them are going to be on the mucous membranes. They're secreted as IgA. And they can stay there for a long time. So if you've got mucosal immunity, okay, the virus, when it arrives to infect you, it gets blocked before it even infects the first cell. So that's why this can be very, very effective. Okay. Okay, one of the things to bear in mind is that not all antibodies that are on the surface of a virus particle are neutralizing. Some of them they can bind, but they won't prevent the virus from infecting a host cell. Okay, so how do we test to see whether we've got neutralizing antibodies present? So you get somebody's serum and you prepare a series of dilutions of this serum from 1 in 4, 1 in 8, and so on. And so we put these dilutions of serum onto cells, and then we add virus at the same dose everywhere through this plate. Then we put the whole thing in the incubator, and then we look, and we've realized that sometimes we've got cells that are infected, sometimes we've got cells that are not infected. So when the serum is present at a high concentration, it will block the infection because it's bound to the surface of these viruses and prevented them from infecting the cells. But when the serum gets diluted, well, in the end, we got like all of our wells get infected. So we got something a little bit like a TCID 50. So we'll determine the neutralizing titer as the dilution that blocks 50% of infections. So we get a graph like this. And here our neutralizing titer will be 1 in 40. So that's basically how seroneutralization works. And if you've got a reporter gene in your virus, then you can do something like this, and you can count the number of fluorescent cells. These are fluorescent cells. These are the ones that have been infected. And here I've got some results in triplicates of cells that have been incubated with pseudotype and also with different dilutions of 2 serums. So this one, whatever the serum dilution, the cells are still getting infected. But here, with our serum with antibodies present, blocks all the infection, some cells getting infected, a bit more, and then all cells getting infected. So our neutralizing titer here will be about 1 in 10,000. Okay, so that's how you test seroneutralization. And if you want to determine whether 2 viruses are different serotypes, firstly, you'll get, for example, the serum from an individual who's had an infection by the strain A virus. So you get their serum, you make up the dilutions, and you put it in 2 different 96-well plates. So in this one, you add the virus strain A. This one, you add the virus strain B. So here, our serum will neutralize the strain A at a certain dilution. But for strain B, even if you've got serum present at only diluted 1 in 4, all these wells are getting infected. So there's no neutralization of strain B by this serum. So that means they're different serotypes. Get infected by strain A, you get antibodies that block the infection of the virus strain A, but they don't block the infection by the virus strain B. And we're talking about the same species of virus, okay? Rhinovirus, adenovirus. It's not 2 different viruses, it's the same species. Okay, so I'm going to ask you a couple of questions here. Think about this for a couple of minutes, and then I will and there are a couple of trick questions in there. Okay, so we'll start from the bottom. Okay, so D, neutralizing antibodies protect against future infection. Who says yes? Yeah, you're right. That's the main importance. C, 1 in 100,000 is a high titer for a neutralizing antibody. Who says yes? Who says no? Okay, that's the reason why I asked this question. So C, the answer is yes. What does this mean? If the neutralizing titer is 1 in 100,000, that means you dilute your serum 100,000 times and it still can block the virus. Okay? If your neutralizing titer is 1 in 100, it means you dilute it 100 times, it will block the virus. But if you go and dilute it 1,000 times, it's not working anymore. So 1 in 100,000 is higher. You've got more neutralizing antibodies in that serum because you can dilute it further and it still has an effect. Okay, so that's the way this scale works, okay? The higher this number, the higher the titer, okay? Because it sounds like it should be the other way around. That's why I put the question in there. About B, neutralizing antibody must prevent the virus from attaching to its cellular receptor. Who says yes? A couple of people who say no. The no is correct. Trick question, because a couple of slides ago, receptor or co-receptor must prevent binding, no, or it could cellular receptor if you prevent binding to the co-receptor or if you prevent activation of the fusion domains. These are slightly different mechanisms. So there's more than one mechanism, all right? And question A, bind to the capsid of naked non-enveloped enveloped virus. Who says true? Who says not true? It's not true. Think about it. Envelope viruses are like this, right? The glycoprotein is what is attaching to the host cell and causing infectious entry. So on an envelope virus, the neutralizing antibodies are going to bind here, okay? They're not going to bind the capsid because that's inside, okay? So A was also a trick question. Yes? Okay, so for example, why is B not correct? So you have to think about the process of entry. So you've got receptor, and here's your attachment glycoprotein. Step 1 is binding. Often, step 2 might be internalization, and then you've got to have this triggering of the fusion domain. So that's binding. 2, triggering of the fusion domain. And then you get infectious entry. And sometimes you might need to have co-receptor. And that will lead to, you might have to go all these 3 steps, one after the other. So question 2 is saying a neutralizing antibody has to block step 1. But that's not true, because if you've got a neutralizing antibody that can block step 2, interaction with the co-receptor, or can block step 3, the triggering in the fusion domain, then that will still prevent the virus from infecting the cell. Because for the infection to work, 1, 2, and 3 have to happen. 1 is the first step, but if you block here or you block here, the virus will still be neutralized. Okay, that's why proposition B is not correct. Yeah, that's why it says must prevent. That's the part of the question that is telling you it's like the only way. It must. Okay, so maybe there's another way to propose a question to make it clearer. Okay, so I'll try and hurry along and look at some of these specific infections. So the first one is poliomyelitis. So it's caused by poliovirus, which, as we all know, is a picornavirus, so non-enveloped capsid positive strand RNA virus. It infects us by the gut, so the infection will be in the gastrointestinal mucosa, and the route of infection, contaminated water, basically, okay, because it's going to replicate in the gut and be excreted in the feces. So probably 95% of infections are asymptomatic. It does not cause gastroenteritis, but it will replicate and the virus will be excreted. However, in maybe 5% of infections, after a 1 to 2 week incubation period, some people infected will have minor symptoms, tiredness, feeling bad, maybe have a bit of a fever that will last for 1 week, and then either this will, you get better, or it can proceed to a meningitic phase. So then you have like headaches, stiff neck, and this means that the virus has started getting into your nervous system. And poliovirus can do this because the receptor it uses is also present on neurons. And then this can get worse, and it may be in less than 1% of total infections. Then there can be acute flaccid paralysis, which can lead to the death of the people because it will paralyze you from the lower members first and then going up. So when the virus infection attacks the neurons that are controlling your diaphragm, you can't breathe anymore and you die. And the people who survive, they will have permanent long-term effects, and that will be 90 to 95% of the people who have been paralyzed. So one of the things we need to know about this is that there are 3 serotypes of the virus. Okay, so fortunately we don't have any polio cases, I think, in France or perhaps even in the whole of Europe. And this is because vaccination was introduced from the middle of the 1950s. So we can see 2 examples of the number of polio cases and deaths, a lower number in the US and in Japan. And this like rising incidence here, and then coming down to 0 after the introduction of the vaccines. So this is a real success of vaccination. However, so what are the vaccines? Okay, there are 2 types of polio vaccines. There are injectable polio vaccine, IPV, and oral polio vaccine, OPV. So one thing I should ask you is, in immunology, have you seen stuff like this, the different types of vaccines in immunology? Huh? Last year, okay. So when this inactivated, attenuated idea, this is new for you or not new? Not new, okay, right. So this is, you get the virus, you inactivate it with formol, and then you inject it, okay? And these ones are attenuated strains, which are viruses that can replicate, but they've lost their their neurotropism and their pathogenicity. So IPV, administered by injection. You need somebody qualified to perform the intramuscular injection, but they're very, very safe, okay, because there's no viable virus here. Oral polio vaccine, you don't need somebody qualified. You can just give it on a drop on a little child's mouth, and the overall cost is much cheaper. Now, the problem occurs because the vaccine strain can actually replicate in the gut, okay? And these vaccine strains will be excreted in the feces of the child who's had the vaccine. Okay, you might say that's not a particular problem, but the problem is that these vaccine strains have acquired mutations that reduce their virulence. But when the virus replicates in the gut of a vaccinated person, then random mutations during the replication cycles can revert the virus back to its pathogenic form. Now, in the person or the child that gets vaccinated, that doesn't cause a problem because their immune system has got an advance on the virus. So it's not a risk for the person who gets the vaccine. But these revertant strains can be excreted by someone who's had the vaccine, and then they can go and infect somebody in the same town or in the same village or in the same family. That's the risk. So what are the mutations? Where are the mutations that cause attenuation here? So there are 3 serotypes of poliovirus. So there were 3 attenuated vaccine strains developed in the 1950s. And since then, we've sequenced them and we've tested them to find out where are the mutations that cause attenuation. So here they are for the serotype 1, serotype 2, and serotype 3 vaccine strains. And from a fundamental immunology point of view, what's interesting is that there are 2 sites that are responsible for attenuation. First of them, firstly, there are mutations in the IRES, the internal ribosome entry site. So these viruses, the translation of their RNAs is less efficient because there are mutations in the IRES. And the second site for attenuation are mutations in the capsid proteins. So perhaps reducing the stability of the capsid or reducing its ability to bind to the poliovirus receptor. But, okay, there are only a few of them, and if these mutations revert, then the virus will recover its virulence, and this will give you what's called VDPV, vaccine-derived poliovirus. And if this VDPV begins to circulate in this population, that's what's called a circulating vaccine-derived poliovirus, CVDPV. And which one of these is going to be the biggest problem? Which vaccine strain has the fewest mutations needed for the virus to become virulent again? Yeah, no. Yes, 2, right? Because we've only got 2 sites here. So if the virus, just by random mutation, manages to switch this one and switch this one, it will become virulent again. Number 1 is better, it's safer. Okay. Number 3, okay, it's got 3 sites, but it's still a little bit better. So it's serotype 2 is going to be the big problem. So when we look at where we have polio still occurring in the world, there's only 1 wild-type poliovirus still in circulation. That's serotype 1, which is only found still endemic in Pakistan and Afghanistan. But in other countries, the burden of disease is from these circulating VDPVs. and specifically VDPV2, okay? Like way more cases of polio caused by this vaccine-derived strain. And it's in a whole swathe of countries across, well, kind of like central northern Africa here, okay? So that's the real problem for polio eradication now, is the ability of these OPV strains to be transmitted and regain virulence. So that's a real issue. So how are we going to try and eradicate this virus? How would we even know? Well, the first thing, you've got to like detect the cases of children with the acute flaccid paralysis. And then you get a stool sample from them, and then you detect the virus specifically by PCR. So this is the global poliovirus laboratory network. So there are global network of labs that are specialized for this. And then you can say, okay, this paralysis is caused by the poliovirus. The trouble with this is less than 1% of infections will give paralysis. Okay, so if you get one case identified like this, there's 200 more infections somewhere in the community. So for community monitoring, then wastewater PCR will be useful to know if you've got poliovirus somewhere, even though there's no symptoms. So rather surprisingly, so this is also done in Europe, and rather surprisingly, we do have some circulating poliovirus in Europe, okay? Even though there are no clinical cases. So every yellow square here, this is the detection of a rather unusual VDPV2, mostly in Germany, but also a little bit in Poland, somewhere in other European countries. And this one is actually a recombinant between the vaccine-derived strain and two other enteroviruses. So this is another issue that's going to really be a problem for eradication of the virus, the capacity of poliovirus to recombine with circulating enteroviruses. And we don't really know how much of a pathogenic potential this recombinant has, okay? Okay, so I think I might come back to this because we have to move on in time. I'm a little bit worried we're going to run out of time, and I think I need to really finish today. So our second infection we're talking about is dengue virus. So we've also seen this one. It's an orthoflavivirus, once again, plus strand RNA virus, this time with an enveloped particle. It's transmitted by a mosquito bite, by Aedes aegyptus or Aedes albopictus, the, I don't know, tiger mosquito, I guess you call it in French. So similarly, there are a number of asymptomatic infections. Not so easy to know how many there are in total. I've looked for the numbers on this. It's not so easy to know. But in a certain number of people, you'll have an incubation period, maybe about 1 week, then a really high fever, headache, and severe muscle pain. Okay, it can be very painful and unpleasant. So this will last several days, and then most people will recover, but other people will have a very severe form, which used to be called dengue hemorrhagic fever. Now the official term is just severe dengue. And we think this is about 1% of all infections, 1%, maybe 0.5%. Now, whether you recover or you die from this infection depends on your access to medical care. So if you are just lying at home suffering from this, you'll probably have like 1 in 5 people who will die. If you have good hospital care, the death rate here will be down to 1%. Okay. And so the burden of disease worldwide is quite a lot. So last year there's estimated to have been almost 50 million cases and more than 12,000 deaths worldwide. So it's a real worldwide health problem. This, will never be eradicated by vaccination because there is an animal reservoir. So even if we were to prevent all human infections, there would still be infected mosquitoes going and biting people and transmitting the virus. So eradication is not an objective here. But the objective for vaccine development is to try and prevent these really, really severe forms that are what causes a lot of illness and is killing people. Now, the problem is there are 4 serotypes of the virus, and there is something very, very specific in dengue infection, which is if you get first infection, serotype 1, then you get a second infection, serotype 2. Not only will you not have protection because of your first infection, the symptoms of the second infection can be worse. They can be more severe. And the thinking currently is that this is due to what's called antibody-dependent enhancement. So if you've got like a dengue virus serotype 1, dengue virus serotype 2, you develop, if you've got your serotype 1 infection, you develop antibodies against this guy. And this will be a mixture of neutralizing and non-neutralizing antibodies. The neutralizing antibodies won't react with dengue virus serotype 2, but some of the non-neutralizing antibodies, they're going to bind here. So they won't prevent the virus infecting cells, but in fact they can be used by monocytes, macrophages that have Fc gamma receptors. They can be used for these cells to bind the virus and get infected by the virus. So there's this phenomenon specific to dengue infection, which is called antibody-dependent enhancement, which leads to this observation that the second infection can be worse than the first. Okay, so those are the kind of like issues with vaccine development. Nevertheless, there have been 2 companies working a lot on this. So first one was Sanofi Pasteur, and they both developed recombinant viruses. So Sanofi Pasteur, their idea was that dengue virus and yellow fever virus, they're both orthoflaviviruses, same type of genome structure, and they kind of work the same way. And we have a good vaccine against yellow fever. So what we got to do is we take the backbone of the yellow fever vaccine strain and we just put the structural proteins, the envelope and the matrix protein of our dengue virus strains into the yellow fever vaccine. So this was tested. There were very good results in 3 major clinical trials. It was authorized, but reanalyzing the results showed that it was already, HIV, no, not HIV positive, dengue positive. Okay, so the only people who had real protection were the ones who had already had a previous infection. And in seronegative children, they actually had a higher risk of symptomatic infection. So the vaccination was doing worse than doing nothing in this population, the very one you want to protect. So this was a kind of huge disaster for the whole field. And the vaccine is maybe still authorized in some situations, but has not been widely used. So currently the best candidate is this one, the TAG003. It's the same kind of idea, but they're basing their vaccine on a recombinant, attenuated type 2 dengue virus. So way back in the 1980s, there were like 53 passages of the virus in canine cells, and they managed to get an attenuated strain of dengue virus. And the idea is we get this attenuated strain and switch out the envelope glycoproteins for the other ones. So the results here are actually more positive. So there were not any more serious infections in the children or the people, children who got the vaccine compared to the non-vaccinated children. So this is more encouraging results reported just last year. Okay, we've got 20 minutes to cover influenza and prion diseases, so I better get a move on. So poliovirus, gastrointestinal infection, waterborne transmission, dengue virus, vector transmitted, influenza, respiratory transmission. Okay, and what happens here is if you get infected, maybe 2 days later, on average, you'll have fever, fatigue, muscle pain, headache, maybe a cough. And you'll feel bad for several days, might feel really tired for maybe up to 2 weeks. But in most people, that's it, you've had the flu. There can be serious forms caused by bacterial co-infection, and also you could get a viral bronchitis or pneumonia. Now, overall, maybe 30 to 50% of infections are asymptomatic in adults less than 65 years old, and the people who are most at risk for these severe infections are elderly people and young children. So overall, as we saw in France from 2018-2019, the case fatality ratio less than 1% for a seasonal influenza virus, and the IFR will be at least 10 times smaller than that. Okay, so the viruses that infect us every year that are circulating are 2 types of influenza A and 2 types of influenza B. Okay, so these H1N1 and H3N2, they are related to the serotypes of the surface glycoproteins. So the surface glycoproteins are the hemagglutinin, responsible for binding and entry, and the neuraminidase, responsible for effective virus release. And it's the antibodies against HA that are the most powerful neutralizing antibodies. So H3N2, these are the serotypes of the hemagglutinin and the neuraminidase. Now, the thing about influenza serotypes is they're more like amorphous serogroups. So if I've got a whole bunch of influenza strains and a whole bunch of sera from people who've recovered from infection, my blue serum here might neutralize strains A and B, but it doesn't neutralize any of the rest. My dark blue serum might neutralize strains B, C, and D, and the light cyan serum might neutralize strains D, E, and F. And then I come along and I look at more sera, and the picture we get is that there's some kind of overlap in the neutralization between all of these strains, and some overlap between these ones, but between them, there's no overlap. So all of these ones will be, for example, serotype H1, and these ones will be serotype H2. But if you look at any one individual serum here, it can't neutralize all of these viruses from the H1 group. Okay, so it's not the same as the dengue or the poliovirus serotypes. So there's a lot of variability in neutralization, and why this happens is because this is the hemagglutinin monomer, the receptor binding pocket is here, and around that, these are the amino acid side chains that can be mutated from one year to the next. So the virus can actually mutate these residues, escape from antibody binding, but it can still bind to a receptor, so it can still infect you. And this is what leads to the process of what's called antigenic drift. So from year to year, these mutations in the hemagglutinin accumulate, and that means that, you know, the virus that's circulating now, 2025, the hemagglutinin is not the same as in the viruses that were circulating 15 or 20 years ago. So that's the reason that the vaccine has got to be updated regularly. And it's also the reason, one of the reasons why this virus will never be eradicated. Now, the other thing that influenza virus is capable of is what's called antigenic shift or antigenic substitution. And this can occur because if you get 2 influenza viruses infecting the same cell in the same person, then both of these virus genomes will replicate in the same cell. And when the virus is released, then a single particle can package some genomic segments from the human virus and other genomic segments from the avian virus. And this occurs, or it can occur, because the genome is segmented. Okay? So if this happens, what you can get is the appearance of a virus that will perhaps have a hemagglutinin from an avian virus. So it will be entirely new to the whole human population. No one has been infected by this virus ever before, but it will have like some human genome segments or human virus genome segments, which means it can replicate effectively in mammalian cells. So whenever this has happened, it's resulted in a pandemic wave of infections. So a new virus would emerge and it can infect everybody because no one has seen this virus before. So most of the pandemics over the 20th century were caused by this kind of reassortment of genome segments, except for Spanish influenza in 1918, which was a direct transmission and adaptation of an avian virus. So because we know this is a real risk, then there's continuing surveillance of avian influenza viruses. Because if they manage to infect somebody who's also got infected by a human influenza virus, then that person could produce a new pandemic virus, okay? So the ones that we've been watching are H5N1, H7N7, H5N8. And currently it's H5N1, which is the most, worrying. And up until 2020, 2019, it was kind of seemed like it was dying out and being replaced by H5N8 in bird populations. But then suddenly in 2020, H5N1 had a massive resurgence. So what happened here? Well, it appears to have been an event happening in Europe, okay, where what occurred was that there was a H5N8 virus, which was highly pathogenic, which recombined with a low pathogenic avian influenza H5N1. So then we got a new highly pathogenic H5N1, highly transmissible in wild birds. So this new H5N1 went all around the world. For the first time, it got to the American continent, because up until 2020, all these highly pathogenic avian influenza viruses were only found in Asia, Europe, and Africa, because they're transmitted by migrating birds. And most of the migration routes are going, Northern Europe to Africa in the winter, or across Asia, Northern Asia, Southern Asia. And there are not so many migration routes that allow the viruses to be transferred from the Eurasian landmass to North America, for example. But this time it did get there, okay? And it had a huge effect on a lot of wild seabird populations. So this is an island, I think this is the Bass Rock, just off North Berwick. And each, like the white dots here, these are the birds that are nesting on the island. So you can see in 2020, the place was like covered in birds. 2022, a lot of empty space. So a lot of wild bird populations, they crashed by 40, 50, 60, 70% of the population was lost over a period of a couple of years because of this. So, as I said, for the first time, it has got to the American continent, and in the US, there are a lot of poultry farms that have been hit by this avian influenza. 184 million birds infected, had to be destroyed to try and control the transmission of the infection just in the last year. And rather surprisingly, it seems also to be able to infect cows. So fortunately, not very many cases transmitted to humans, but the virus has only got to get lucky once, right? It's got to just get the right set of mutations, and then that's it. It could be like the new COVID-19. Could be some guy working in a farm in California or something, and that's it. This will be the next pandemic. Okay, I've got 10 minutes to cover prion diseases, and I think we will probably manage, be okay. So prion diseases, the last one, and I guess the kind of connection with the previous section is transmission from animals to humans is the link here. So we start off thinking about an animal disease, and this is scrapie, which is a disease in sheep and goats, which has been described since the 18th century. And at the beginning of the 20th century, there started to be a serious scientific interest taken in this disease in veterinary schools. So in France, there was the first demonstration that this disease could actually be transmitted. So we knew by, dissecting sheep that had been very, very severely ill, affected by this disease. You take different organs, and the pathologists have a look at the histology of the different organs in the animal. And you say, okay, this animal has got a brain pathology. Okay, so we know it's like a disease affecting the brain. And once you can see you've got lesions in the brain, then you can get a kind of extract from an infected sheep from their brain and try and introduce it into another sheep. And if you do that, then in fact, the infected, the sheep that have been injected get the same disease. With a slow disease progression, even with an intracerebral injection, it will take 1 to 2 years. But once you've done this, you can test other modes of transmission. And if you get an intraperitoneal injection, then the sheep will still develop disease, but it will take a longer time to develop. So this is how we know this is a transmissible disease. You can transmit from an infected animal to a non-infected animal. And then they did stuff like at the beginning of the 20th century, try and filter it, try and get a culture. We can't culture it, we can't filter it. So it looks like a virus, okay? A filterable, transmissible agent. And then in 1967, there was this very important experiment. They got infectious material from the brain of a sheep with scrapie, zapped it with a huge dose of ionizing radiation, enough to destroy any DNA or RNA, and then injected that. It was still infectious. So what does this mean? That means there's no nucleic acid in the infectious particle. That is a very, very weird thing, right? Well, you don't look very surprised. Good grief. Hang on. So why is this surprising? It's transmissible. It can cause disease. So somehow this thing has got some kind of biological information that allows it to cause disease in another animal. But there's no nucleic acid. That's why it's surprising, because biological information, it's in DNA or it's in RNA, but not here. So what is it? Okay, so there's a human connection here. So this comes from observations of 2 rare encephalopathies in humans, Creutzfeldt-Jakob disease. So this is a fatal neurodegenerative disease. Most cases just sporadic, nobody knows why you get them, but 10% of cases are within families, so they're genetic. And the other one is Kuru, which some people can get or used to be able to get in Papua New Guinea. And this was related by the funeral practices. The people went to the funeral and they got the brain of the dead person and touched it, okay, and maybe like even 8 bits of it or something. So, and then you had this very, very unusual funeral rite which related to transmission of the disease. And in both cases, there were these very characteristic spongiform lesions in the brain. And both could be transmitted to chimpanzees in the same way as scrapie. And the first person to realize that there might be a connection here was a vet called Mr. Hadley. And he said, yeah, this Kuru thing looks like it's scrapie. It looks like the same thing as scrapie. And so scrapie, Kuru, and Creutzfeldt-Jakob disease, they are collectively called prion diseases. So no RNA, no DNA. What is the biological information? How do these, what actually is causing this prion disease? So we don't know what it is, but it's somewhere in the brain of these infected sheep. Let's try and purify it. So this was Stanley Prusiner who did this in 1982, was able to get a highly purified, infectious preparation from brains of scrapie infected sheep. And it was a protein called the prion protein, PRP. And this purified protein was infectious. So he was able to get a sequence from the N-terminus of the protein. And then if you've got the protein sequence, then you can get the gene. And this gene was the same one as had been identified in the familial forms of Creutzfeldt-Jakob disease, in the families who had a genetic form of this rare disease. So we've got, in one case, the protein in sheep is transmissible, and in humans, if it's mutated and you get it from your parents, it can also cause disease. So what is happening here? What is happening is that this prion protein can exist in two conformations. Normal conformation here, which has got a bit of alpha, of completely alpha helical, and the pathogenic conformation, which has got a large part of the protein in beta sheet. So the normal conformation is called PRPC, and the pathogenic conformation is PRPSC for scrapie. And what is happening is that if the PRPSC interacts with proteins that are in the normal conformation, it will bind to a protein in the normal conformation and then bend it out of shape so that it assumes the pathogenic conformation. And this process continues with the formation of aggregates of the PRPSC. So these aggregates will build up in the brain, and they are what are causing the neuropathy. So what is the biological information? It's the three-dimensional conformation of the protein, which somehow has acquired the ability to reproduce itself by interacting with normal versions of the protein. So it's not a sequence of DNA or RNA or even the sequence of the protein. It's the three-dimensional conformation of the protein that is being replicated. Okay, so this is a really crazy, unusual thing. Okay, so we got some rare familial disease and we have a sheep disease. Is there any risk for us? So we had an event in the United Kingdom where we, in the 1990s, there was the appearance of a new form of Creutzfeldt-Jakob disease. So the same pathology in the brain, but this time it was young people getting these symptoms and then dying. And it was traced back to or were linked to mad cow disease, bovine spongiform encephalopathy. And where did that come from? So a new disease in cows. Well, it came from a change in the practices by which tissues from sheep with scrapie were converted into like feed supplements to give and given to cows to eat. So we ground up the carcasses of these scrapie infected sheep, turned them into flour, and then gave that to cows to eat. And some of these sheep were infected with scrapie, and the prion protein was able to establish an infection in cows. And because this is a slow disease, it probably took maybe 10 years for this to become apparent. So before anybody knew this was happening, then we had a whole cohort of infected cattle. And some people were eating the beef from these animals, right? And then some of these people got infected. So the prion protein from the BSE infected cows was able to get transmitted to people and turn all of our proteins into the scrapie conformation. So overall, there are about 170 cases over the decade 1996 to 2006. And this is still the reason why I cannot give a blood donation in France, because I lived in the UK during the period of the highest transmission of mad cow disease to humans. So apparently I've got a risk in my blood of giving Creutzfeldt-Jakob disease to somebody. Well, according to the Établissement Français du Sang, in any case. So that's a past event. What about now? Well, there's still a certain zoonotic risk, because there is another naturally occurring prion disease in deer in North America. This is called chronic wasting disease. And in the last 25 years, it has really expanded from a few isolated outbreaks to a large number of, so some of these deer, they're farmed in America for their meat, and there are also wide-ranging populations. So there's a lot of wide-ranging and farm populations that have been infected with this prion. And of course, people go out and hunt them and eat them, and they won't know if they're shooting an animal that's got prion disease but hasn't really developed symptoms yet. So there's a bit of a question of whether chronic wasting disease will ever spill over into the human population. So that's about it. So the things that I wanted to cover today is, if you get a acute virus infection, you'll have a diverse spectrum of pathologies, from asymptomatic to fatal. That's between different infections and also within the same virus infection, okay? Not everybody who gets a dengue virus infection will have a severe infection. Most people will have a mild infection, and only certainly a minority of people will have a severe infection and die. So these numbers, CFR, EFR incidence prevalence, important. Whether we can get reinfected by the same virus or whether vaccination can really be effective depends on this neutralizing serotypes concept. That's really, really important for controlling, eliminating viruses by vaccination. And finally, the weirdest thing in the whole of biology, maybe, prion diseases. It's the conformation of the protein that causes the disease, and it can replicate by folding the other proteins into the same conformation. Okay, and as I said, maybe it's kind of the opposite of a viroid. The viroids in plants, they're infectious RNA without any protein, and a prion is the opposite. It's an infectious protein without any nucleic acid. Okay, so I stop there, and I'm pretty much on time. That's good, right?